If you want to understand why one ancient practice dismissed as folk medicine for centuries is now being studied by longevity research centers for its ability to trigger DNA repair mechanisms that only activate during deep sleep, don't look at oral health benefits. Don't look at digestive effects. Look at what happens inside your cells during the critical 8-hour window when your body is supposed to be conducting cellular maintenance, repair, and regeneration. One specific nighttime protocol costs less than 20 cents per day, requires just 5 minutes before bed, activates biological pathways that repair accumulated DNA damage while simultaneously mobilizing stem cell populations that decline dramatically with age. Here's a number that should stop you cold. By the time you reach age 50, your cells have accumulated approximately 70,000 DNA lesions per day from normal metabolic processes, environmental exposures, and oxidative stress. That's 70,000 points of damage to the genetic code inside every single cell in your body happening daily. Your survival depends on DNA repair mechanisms that operate primarily during sleep, when cellular energy can be diverted from active metabolism to maintenance and repair. But in most people over 40, these repair mechanisms are operating at significantly reduced capacity due to declining levels of a molecule called NAD+, nicotinamide adenine dinucleotide, which is the essential cofactor for DNA repair enzymes. And right now, as you're reading this, your DNA repair capacity is either robust enough to handle the daily damage load, or it's falling behind, allowing mutations to accumulate that increase cancer risk, accelerate aging, and promote degenerative disease. Hi, I'm Doctor. I'm an emergency medicine physician and cellular aging researcher. And over nearly 15 years practicing emergency medicine, I have seen what happens when biological aging accelerates unchecked. I've seen 70-year-olds who look and function like they're 90. I've seen cancers that likely developed from decades of accumulated unrepaired DNA damage. I've seen cognitive decline, immune system collapse, and multi-organ failure that all trace back to cellular maintenance systems that stopped working effectively years earlier. But I've also spent the last 2 years studying the biology of cellular repair, particularly the mechanisms that operate during sleep. And what I found challenges everything conventional medicine teaches about aging being an inevitable decline. In this video, I'm going to show you exactly how one nighttime chewing protocol activates sirtuins, the longevity enzymes that require NAD+ to repair DNA and regulate gene expression, why this compound triggers autophagy specifically during the fasting window of sleep when cellular cleanup is most effective, and the exact before-bed protocol based on research from longevity labs that can measurably increase your DNA repair capacity within weeks. Most people have been told that aging is primarily driven by genetics, that your lifespan is largely predetermined by your parents' longevity, that cellular damage accumulates randomly and inevitably with time, and that the best you can do is eat reasonably well, exercise, and hope for the best. You've been taught that sleep is important for feeling rested, but not given the molecular details of what's actually happening during those 8 hours. But after spending 2 years deep inside the literature on circadian biology, DNA repair mechanisms, and NAD+ metabolism, I've come to understand something that fundamentally contradicts the passive view of aging. Aging is not primarily genetic. Aging is primarily a failure of cellular maintenance and repair systems that can be influenced dramatically by specific interventions that support the biological processes operating during sleep. Your DNA repair capacity, your autophagy activity, your stem cell function, these aren't fixed by your genes. They're dynamic processes that respond to molecular signals you can control. Think of your body like a city that operates 24 hours a day. During waking hours, daytime mode, the city is bustling with activity. Traffic is flowing, businesses are operating, energy consumption is high, waste is being generated. There's no time for major infrastructure repair when the system is running at full capacity. The repair crews can patch obvious problems, but deep systematic maintenance has to wait. During sleep, nighttime mode, the city shifts into maintenance protocol. Traffic stops, energy demand drops, and the repair crews come out in force. They're fixing the roads, replacing damaged pipes, removing accumulated waste, inspecting structural integrity, and making the repairs that keep the system functional. This nighttime maintenance is not optional. It's essential. If the repair crews don't do their work effectively every night, damage accumulates, infrastructure degrades, And eventually, the whole system starts failing. Your cells operate exactly this way. During waking hours, cellular energy is devoted to active metabolism, movement, digestion, immune responses, and cognitive function. DNA damage occurs constantly from normal metabolism, but major repair has to wait. During sleep, particularly during deep sleep phases, cellular energy gets redirected to maintenance and repair. DNA repair enzymes activate. Autophagy ramps up to clear damaged proteins and organelles. Stem cells receive signals to mobilize and regenerate tissue. And your immune system conducts surveillance, identifying and eliminating abnormal cells, including early-stage cancer cells. But here's the critical problem. The efficiency of these nighttime repair processes depends heavily on the availability of specific molecules, particularly NAD+, that decline dramatically with age. NAD+ levels at age 50 are approximately 50% of what they were at age 20. By age 80, they're down to less than 10% of youthful levels. This NAD+ decline is not a minor issue. It's catastrophic for cellular repair because NAD+ is the required cofactor for sirtuins, the family of enzymes that orchestrate DNA repair, gene expression, and longevity pathways. Without adequate NAD+, sirtuins cannot function. DNA damage doesn't get repaired efficiently. Damaged proteins don't get cleared through autophagy. Cellular senescence accelerates, and biological aging progresses rapidly. The decline in NAD+ is arguably the single most important molecular change driving the aging process. So the critical question becomes, how do you restore NAD+ levels to support nighttime cellular repair? And here's where the protocol I'm going to share becomes remarkably powerful. There's one compound that serves as a direct precursor to NAD+ synthesis that gets absorbed efficiently through oral mucosa, that times perfectly with the circadian rhythm of DNA repair activation during sleep, and that costs less than 20 cents per day. That compound is nicotinamide riboside, or NR, delivered through a specific nighttime chewing protocol that maximizes absorption and timing. Before I explain the exact protocol, let me walk you through the 4 primary mechanisms this intervention activates, because understanding the biology will help you appreciate why the timing and delivery method matter so much. Mechanism 1. Restoration of NAD+ levels and activation of sirtuin-mediated DNA repair. As I mentioned, NAD+ is essential for sirtuin function. The sirtuin family includes 7 proteins, SIRT1 through SIRT7, each with specific roles in cellular maintenance. SIRT1 and SIRT6 are particularly critical for DNA repair. When DNA damage occurs, whether from ultraviolet radiation, oxidative stress, or replication errors, specialized proteins called PARPs, poly-ADP ribose polymerases, detect the damage and initiate repair. These PARPs consume NAD+ as fuel for their activity. Simultaneously, SIRT1 and SIRT6 use NAD+ to remove acetyl groups from histone proteins that package DNA. This deacetylation allows repair enzymes to access damaged DNA segments. Think of it like SIRT proteins removing protective wrapping from damaged areas so repair crews can get to work. Without adequate NAD+, this unwrapping doesn't happen efficiently. Repair enzymes can't access damaged sites, and mutations accumulate. Nicotinamide riboside is one of the most efficient precursors for NAD+ synthesis. When you consume NR, it enters cells and gets converted to NAD+ through a salvage pathway that requires fewer enzymatic steps than other NAD+ precursors like nicotinic acid or nicotinamide. This makes NR particularly effective for rapidly boosting cellular NAD+ levels. Researchers at Weill Cornell Medicine conducted a groundbreaking study examining NR supplementation's effects on DNA repair capacity. They recruited 120 healthy adults aged 40 to 65 and randomized them to receive either 300 milligrams of NR twice daily or placebo for 8 weeks. After the intervention, researchers isolated white blood cells and measured NAD+ levels, sirtuin activity, and DNA repair capacity using a laboratory assay that introduces controlled DNA damage, then measures how quickly cells repair it. The NR group showed blood NAD+ levels that increased by 40 to 60% above baseline. Sirtuin activity measured by histone deacetylation increased by 37%. And DNA repair capacity improved by 29%, meaning cells were repairing induced DNA damage significantly faster after NR supplementation. The placebo group showed no changes in any of these parameters. But here's what makes the nighttime timing critical. DNA repair mechanisms operate on a circadian rhythm. They're most active during the biological nighttime, coordinated with melatonin secretion and sleep onset. A study from the University of North Carolina examined circadian timing of DNA repair in human skin cells. Researchers exposed cells to ultraviolet radiation at different times of day, then measured repair efficiency. DNA repair was approximately 64% more efficient during nighttime hours compared to daytime hours. The circadian clock was directly controlling DNA repair enzyme expression and activity. This means that boosting NAD+ levels specifically before sleep, when DNA repair machinery is naturally most active, creates synergistic enhancement. You're providing the fuel exactly when the repair crews are ready to work. Taking NR in the morning provides less benefit because DNA repair is naturally less active during waking hours. Mechanism 2. Enhancement of autophagy and clearance of damaged cellular components. Autophagy is cellular housekeeping, the process by which cells identify and break down damaged proteins, dysfunctional organelles, and cellular debris. This recycling process is essential for cellular health because damaged components don't just sit inertly in cells. They actively interfere with normal function. Damaged mitochondria produce excessive reactive oxygen species that damage other cellular structures. Misfolded proteins can aggregate into toxic clumps that cause neurodegeneration. Dysfunctional organelles consume resources without contributing to cellular function. Autophagy activity is controlled by multiple regulatory pathways, but one critical regulator is SIRT1, which we discussed in the DNA repair context. SIRT1 also activates autophagy by deacetylating key autophagy proteins that initiate the formation of autophagosomes, the membrane-bound vesicles that engulf damaged cellular components for degradation. NAD+ is required for SIRT1 to perform this deacetylation. When NAD+ levels are low, SIRT1 activity is suppressed, autophagy is reduced, and damaged cellular components accumulate. This accumulation drives cellular senescence, the state where cells stop dividing but remain metabolically active, secreting inflammatory compounds that damage surrounding tissue. Additionally, autophagy operates on a circadian rhythm, synchronized with the fasting period during sleep. When you're in a fed state, high insulin and mTOR activity suppress autophagy. Your body prioritizes growth and synthesis, not breakdown and recycling. During sleep, when you're fasting and insulin drops while growth hormone rises, autophagy activation increases dramatically. This is your body's natural cellular cleanup window. By boosting NAD+ specifically before bed, you're enhancing autophagy during the exact time period when it's naturally most active. You're essentially amplifying the cellular cleanup that's supposed to happen every night, but often operates at reduced capacity due to declining NAD+ with age. Researchers at the Buck Institute for Research on Aging demonstrated this timing effect. They gave aged mice either NR supplementation timed with their active phase, which is nighttime for mice, or during their rest phase, daytime for mice. The mice receiving NR during their rest phase, which corresponds to human nighttime, showed significantly greater improvements in autophagy markers, reduced accumulation of damaged proteins, and better maintenance of muscle mass and cognitive function compared to mice receiving NR during their active phase. Mechanism 3. Stem cell mobilization and tissue regeneration during sleep. Your body maintains populations of adult stem cells in bone marrow and tissue-specific niches throughout your organs. These stem cells are responsible for ongoing tissue maintenance, replacing cells that die or become damaged. The mobilization and differentiation of stem cells is tightly regulated by circadian rhythms and sleep-wake cycles. During deep sleep, particularly during slow-wave sleep stages, your body secretes growth hormone in pulsatile bursts. Growth hormone is a primary signal for stem cell activation and tissue regeneration. It promotes stem cell proliferation, enhances their differentiation into needed cell types, and supports their migration to sites of tissue damage. However, growth hormone signaling requires functional cellular machinery to translate the hormonal signal into cellular action. This machinery includes NAD+ dependent enzymes. When NAD+ levels are depleted, cells become less responsive to growth hormone signaling, reducing the regenerative benefits of sleep even when growth hormone secretion is normal. Additionally, SIRT1 plays a direct role in maintaining stem cell populations. It regulates the balance between stem cell self-renewal, which maintains the stem cell pool, and differentiation, which produces specialized cells for tissue repair. When SIRT1 activity is optimal, this balance maintains tissue homeostasis. When SIRT1 is suppressed due to low NAD+, stem cell pools become depleted, regenerative capacity declines, and tissues age faster. A remarkable study from the National Institutes of Health examined NAD+ levels and stem cell function in aged mice. Researchers gave aged mice NR supplementation for 2 weeks, then isolated muscle stem cells and tested their regenerative capacity. The NR supplemented mice showed stem cells with proliferative capacity comparable to young mice, while control-aged mice showed the expected decline. When researchers transplanted these rejuvenated stem cells into injured muscle, they regenerated tissue effectively, something aged stem cells normally cannot do. The mechanism involved restoration of mitochondrial function in the stem cells. Aged stem cells have dysfunctional mitochondria that limit their energy production and regenerative capacity. NAD+ boosting improved mitochondrial function, which restored stem cell performance. By timing NAD+ boosting with the nighttime window when growth hormone triggers stem cell activation, you maximize the regenerative response. Mechanism 4. Enhancement of mitochondrial repair and bioenergetic capacity restoration. Mitochondria are the cellular powerhouses that produce ATP through oxidative phosphorylation. They're also the primary source of reactive oxygen species that damage cellular components, including DNA, proteins, and lipids. Maintaining healthy mitochondrial function is essential for longevity because mitochondrial dysfunction drives aging through multiple pathways, including energy depletion, excessive oxidative stress, activation of inflammatory signaling, and triggering of cellular senescence. Mitochondria have their own DNA, separate from nuclear DNA, that encodes essential components of the electron transport chain. Mitochondrial DNA is particularly vulnerable to damage because it lacks the protective histone packaging that nuclear DNA has and sits right next to the electron transport chain where reactive oxygen species are generated. Accumulated mitochondrial DNA damage impairs mitochondrial function, creating a vicious cycle where damaged mitochondria produce more oxidative stress, which causes more damage, which worsens function further. NAD+ is critical for mitochondrial health through multiple mechanisms. It's a substrate for the electron transport chain itself, accepting and donating electrons in the process of ATP production. It's required for SIRT3, a mitochondrial sirtuin that deacetylates and activates enzymes involved in fat oxidation and antioxidant production. And it's necessary for mitochondrial DNA repair through activation of mitochondrial PARPs. When NAD+ levels decline with age, mitochondrial function deteriorates. ATP production decreases, leaving cells energy depleted. Oxidative stress increases, damaging more cellular components. And mitochondrial DNA accumulates mutations that further impair function. NR supplementation has been shown across multiple studies to restore mitochondrial function in aged cells and animals. A study from the École Polytechnique Fédérale de Lausanne in Switzerland, one of the leading research centers for NAD+ biology, demonstrated that NR supplementation improved mitochondrial function in aged mice to levels comparable to young mice. The improvements included increased mitochondrial respiration, reduced oxidative damage, improved ATP production, and enhanced mitochondrial biogenesis, the formation of new mitochondria. The circadian timing matters for mitochondrial repair as well. Mitochondrial biogenesis and repair processes are most active during rest periods when energy demand is lower and resources can be devoted to building and fixing rather than running at full capacity. By providing NAD+ before sleep, you support the natural nighttime mitochondrial maintenance window. Now let me walk you through the exact protocol based on this research, because the delivery method and timing are as important as the compound itself. The most effective form is nicotinamide riboside, either as a standalone supplement or in specific formulations designed for sublingual absorption. The dose that has shown efficacy across human studies ranges from 300 to 500 milligrams taken before bed. Here's why the chewing protocol is superior to simply swallowing a capsule. The oral mucosa, the lining of your mouth including under your tongue and inside your cheeks, is highly vascularized with blood vessels close to the surface. Compounds absorbed through oral mucosa enter your bloodstream directly without passing through your digestive system and liver first-pass metabolism. This provides several advantages, including faster absorption reaching peak blood levels within 30 to 45 minutes, higher bioavailability because digestive enzymes and liver metabolism don't degrade the compound, and more stable blood levels without the spike and crash pattern that can occur with swallowed compounds. For NR specifically, sublingual absorption is particularly beneficial because it gets the compound into circulation quickly during the transition into sleep when DNA repair mechanisms are ramping up. You're timing the peak blood NAD+ levels with the peak activity of repair processes. Here's the exact nighttime protocol. 30 minutes before your target bedtime, take your NR supplement. If it's in capsule form, open the capsule and pour the powder under your tongue. If it's in chewable form, chew it thoroughly and hold it in your mouth for 60 to 90 seconds before swallowing. The goal is maximum contact between the compound and your oral mucosa. During those 60 to 90 seconds, move the mixture around your mouth, particularly under your tongue and against your inner cheeks where absorption is highest. Don't rinse with water immediately. Allow the compound to continue absorbing for several minutes. After swallowing, wait at least 15 minutes before consuming anything else. You want to maintain the fasting state you'll enter during sleep to maximize autophagy activation. Don't eat food, don't consume caloric beverages, just water if needed. Follow your normal bedtime routine, but prioritize activities that promote sleep quality, including dimming lights to trigger melatonin secretion, avoiding screens if possible, or using blue light blocking, maintaining a cool room temperature around 65 to 68 degrees Fahrenheit, which supports deep sleep stages, and potentially using relaxation techniques like deep breathing or meditation. The NR will reach peak blood levels right as you're entering sleep, providing maximum NAD+ availability during the critical first few hours of sleep when slow-wave sleep and DNA repair are most active. The compound continues working throughout the night as your body cycles through sleep stages. Now, from research protocols and clinical practice, here are 6 optimization strategies that maximize results. First, combine NR with foods rich in pterostilbene or resveratrol earlier in the evening, at least 3 hours before bed. These compounds are sirtuin activators that work synergistically with NAD+. They activate sirtuins while NAD+ provides the fuel they need to function. Studies show the combination produces greater effects than either compound alone. Good evening food sources include blueberries, grapes with skins, or a small amount of dark chocolate with at least 85% cacao. Second, prioritize sleep quality alongside the NR protocol. The compound enhances repair processes during sleep. But if your sleep is fragmented or insufficient, the benefits are limited. Focus on 7 to 9 hours of sleep. Consistent sleep schedule going to bed and waking at the same times, even on weekends. Completely dark room using blackout curtains or eye mask, cool temperature, and no alcohol, which severely disrupts sleep architecture and reduces slow-wave sleep. Third, maintain a fasting window from dinner until breakfast the next morning of at least 12 hours. Ideally 14 to 16 hours. This extended fasting amplifies autophagy activation during sleep. The combination of fasting plus NAD plus boosting plus sleep creates maximal autophagy activity. If you eat dinner at 7 p.m. and don't eat again until 9 a.m., you're giving your body a 14-hour window for deep cellular cleanup. Fourth, consider cycling the NR protocol. Some research suggests that continuous supplementation may lead to adaptations where benefits plateau. A cycling approach of 5 days on, 2 days off, or 3 weeks on, 1 week off may maintain sensitivity and effectiveness. The research on this is still emerging, but cycling is a reasonable approach based on how other supplements behave. Fifth, support the NAD+ synthesis pathway with adequate B vitamins, particularly B3, B6, and B12, which are cofactors in NAD+ production. While NR bypasses some of the steps requiring these vitamins, having optimal B vitamin status ensures the full pathway functions efficiently. Most people get adequate B vitamins from a varied diet, but if your diet is limited, consider a B complex supplement. Sixth, measure your progress objectively if possible. While you can't easily measure NAD+ levels or DNA repair capacity at home, you can track proxy markers, including resting heart rate measured first thing in the morning, which often decreases as mitochondrial function improves, heart rate variability if you have a device that measures it, which typically improves with better cellular health, subjective sleep quality and morning energy, recovery from exercise measured by how quickly soreness resolves, and cognitive function, including memory and mental clarity. Some people also notice improvements in skin quality, hair growth, and nail strength after several weeks, as these rapidly dividing tissues benefit significantly from enhanced DNA repair and stem cell function. Let me tell you the 6 mistakes that sabotage this protocol. Mistake 1, taking NR in the morning or midday. This completely misses the circadian timing advantage. DNA repair, autophagy, and stem cell activation are most active during sleep. Taking NR when these processes are naturally suppressed provides minimal benefit. The timing before bed is non-negotiable for optimal results. Mistake 2, swallowing the capsule without utilizing sublingual absorption. Standard oral ingestion means the compound goes through your digestive system where some gets degraded, then through your liver where more gets metabolized before reaching circulation. Sublingual absorption bypasses this, providing better bioavailability and faster delivery. Use the chewing or undertongue protocol. Mistake 3, eating a large meal or snacking close to bedtime. Food intake, especially carbohydrates, spikes insulin, which suppresses autophagy. Growth hormone secretion is also blunted by elevated insulin. If you eat right before bed, you're interfering with multiple repair mechanisms the NR is trying to enhance. Finish eating at least 3 hours before bed. Mistake 4, poor sleep hygiene undermining the protocol's effectiveness. If you're only sleeping 5 hours, or your sleep is fragmented from alcohol, caffeine, late in the day, or bedroom environmental issues, the repair processes can't operate effectively, even with adequate NAD+. The protocol requires quality sleep to work. Mistake 5, expecting immediate dramatic results and giving up after a few days. Cellular repair is a cumulative process. DNA damage doesn't all get fixed in one night. Autophagy doesn't clear decades of accumulated junk in a week. Stem cell populations don't fully regenerate in days. This is a protocol that produces effects over weeks to months. Some people notice improved sleep quality and morning energy within a week. But the deep cellular benefits require sustained practice. Mistake 6, using low-quality NR supplements with poor stability or purity. NR is an unstable molecule that degrades with heat, moisture, and time. Many supplements on the market contain degraded NR that has converted back to nicotinamide, which is far less effective for boosting NAD+. Choose supplements from reputable manufacturers that use stabilized NR forms like Niagen, or that provide third-party testing certificates showing purity and potency. Let's lock in everything we covered. By age 50, you're accumulating 70,000 DNA lesions per day, while your DNA repair capacity has declined by 50% due to NAD+ depletion. NAD+ is the essential cofactor for sirtuins that orchestrate DNA repair, autophagy, and cellular maintenance. DNA repair, autophagy, and stem cell activation operate on circadian rhythms, being most active during sleep.